Is slide visible, sir?
Yes, madam.
Okay. So, shall I start the session?
मैडम प्रेजेंटेशन मोड कर लीजिए. और ये मोबाइल डिवाइस आपका है? थैंक यू मैडम.
Now, okay, sir. So, good evening, everyone. Today, I'll be speaking on modern techniques in crop improvement. So, as we all know, that crop improvement is a backbone of agriculture development and food security, right? So, with the increasing population pressure and with this devastating change in climate and shrinkage of the natural resources, traditional approaches of breeding is not alone sufficient for fulfilling whatever is the food, feed, fodder requirement for our emerging population as well as the cattle, right? So modern science has introduced powerful tool which allow us to improve not only the yield but also focus on the quality parameters and and make our crop more resilient towards in an effective, efficient and precise way towards the day by day changing climatic scenario. So in this presentation, I will take you through the evolution of crop improvement, right? Starting from what are the conventional breeding techniques which we are following. Not, I must not say earlier, but till date also, even though we are being a breeder, we can include or go through both the approaches. We can merge with the modern techniques, but we have it's still the hold for our the past old traditional conventional breeding method. So right, starting from the conventional breeding to advanced technology like molecular breeding, genome editing, speed breeding, and nowadays AI, which is a very most important at most thing. And the aim is to highlight how these approaches, right, collectively contribute towards gaining the sustainability towards the agriculture as well as the future food security. So first of all, we will discuss here how the crop improvement, why it is needed, and what is the impact of this crop improvement with respect to crop production and productivity. So crop improvement is very essential, as I told you, to meet the rising demand for food, feed, fodder, fiber, bioenergy. So when this through this technique, through the techniques of different techniques of crop improvement, uh, and with this we can gain or we can get improved variety, which ultimately help farmers to achieve the higher productivity even under stress condition, whatever it may be the stress. Either it may be the biotic stress, which includes pest and disease, or it may be the abiotic stress, say for example drought and salinity. So traditionally the crop improvement relied on phenotypic selection, but advances but with the recent advances in genetics, genomics and biotechnology, that has totally transformed the process what we were following in the past and what we are following today in a recent modern world. So today we can combine both conventional breeding with molecular tools to accelerate either it may be the varietal development. Say for most important thing, crop improvement is not only about the yield. In very first, I told you, it also focuses on the quality parameters. Say for example, nutritional quality, for environmental sustainability and economic viability. By using diverse genetic resources or eco-friendly breeding strategies, modern crop improvement supports both, we can say, productivity as well as environmental conservation. Right. So first and foremost thing, when we talk about the diversity, how diversity will create, or how the variation will create, we all have heard, and we all know that genetic diversity forms the foundation for all crop improvement. Even if you can see your hand, all the five fingers are not same, right? Each and every finger is having, like, irrespective of their you can say the uses and holding grip towards the things whenever you utilize your hand, right? The same diversity before, if the diverse population is present, then only you will go for the selection, right? Or if we want to create the diversity, the variation is required. How you can create a variation among a given base population, right? By earlier we used to follow it by traditional method, but now we have combined it with the modern technique so that we can simplify, or we can cut short the generations. Say for example, in speed breeding, just in 2 years you will be in handed with your product to get released or for the testing in the different pipelines. Either it may be the IVT, AVT, correct? So I told you genetic diversity forms the foundation of all crop improvement program, and germplasm conservation ensures, like, whatever the variability, whatever the material you will select, ultimately the thing you need to conserve it. Okay, that is what whenever you will be having a thought of crop improvement, modernization technique, development of variety, diversity and conservation, these are parallelly two sides of coin which runs together, right? So germplasm conservation mainly ensures with availability of diverse gene, which may be required to tackle the future challenges, whatever it may be, either the urgent surgence of some new pest, disease, or different climatic vagaries or extremities, too hot, too cold, flash flood, intermittent rain fall. So these things. Gene bank, in this regard, gene bank plays a crucial role in preserving the land races, wild relatives, and allied lines. As you might have heard about IVPGR and NVPGR, right? So whenever the breeder requires the material, he or she can write and they can get the material. So biotechnology also support the sustainable agriculture, but by improving the resource use efficiency, how efficiently you are using the, because nowadays we don't have the climate vulnerabilities, we have also scarcity of the land, scarcity of the water resources. So these resources is also sinking day by day. And also we can keep in a mind less and reduced utilization of chemical input. So without conserving genetic diversity, breeding progress would get stagnant. You are not going to get put forward without the conservation of your genetic material along with you. So therefore, integrating germplasm conservation with modern breeding ensure long-term agriculture resilience. Right. So now coming to the different landmark stages, how in AI it comes point 1, 1.0, 2.0, 3.0. Likewise, in breeding, also we have the different landmarks stages, breeding 1.0. So plant breeding has evolved through several landmark stages. So breeding 1.0, this involves simple selection by farmers. Whatever the nature selects, the survival of the fittest, that carry forwards the next generation as the advanced material, right? That is the earliest process. In breeding 2.0, we have introduced Mendelian genetics. We have the plant way. We know how the genes segregate, what is the independent assortment, what is the law of segregation, how the combinations forms, what about the reciprocal crosses, what about the different ratios. Okay, and when we have a plant hybridization program. Now, coming to the stage breeding 3.0, we have started with the mutation part. So mutation is also by chance. We called it as a spontaneous mutation that nature provides us. But in some cases, artificial mutation we can also induce, which involve both physical and chemical mutagens, right? With third step breeding 4. Fourth step breeding 4.0, the marked era of genetic engineering and transgenic crop has been evolved and started. Here the breeding, like how you can study about the gene pool 1, 2, 3 is okay, but in gene pool 4 we are making the crosses apart from the boundaries of genera, speciation, species, right? So here in breeding 4.0, we have introduced our biotechnology to integrate with our conventional breeding methods. And now today, breeding 5.0 uses genome editing techniques, different techniques in genome editing, CRISPR-Cas for precise gene modification. So these progression reflects increasing, you can see we have the methods earlier also, but that is time taking. It is consuming lot of time and precision part is very, very less. But when we included or when we combined our conventional breeding part here, right, is starting from the natural selection to the human selection, artificial selection, and then combining the mutation, carry forward it towards our genetic engineering part, gene editing part. So we are making our crop improvement program more accurate, speedy, as well as efficient, right? So here we can see, I have shown in this diagram how earlier we used to select in a natural condition, in a conventional breeding part. Then we have a knowledge of gene, allele. Then we know this is the parent P1, P2. We are making the F1, how the genes segregate. Then with that, we have started with this breeding 3, that is mutation. Either the nature shows or we chemically induce the mutation to create the variation. And side by side with the genetic transformation, agro-mediated transformation, we have started our gene editing. That is the utmost now today what we are having, the running. You might have been heard about the tilling, eco-tilling, the genomics, phenomics. Now nobody is talking about the simple pure line, mass selection, pedigree, even though that is the backbone. But to speed the breeding procedure, we combine with the modern technology. We are not solely wholly depend upon the modern techniques. So now coming to the conventional breeding method, as I told you, there are different methods like domestication, selection. Okay, so whatever the part you have with you, like we found some wild progenitors that is having some efficient, but they are lacking in what type of the things. Yield is very less, right? So we want to incorporate some of the genes which is present in their gene pool, like they have the biotic, abiotic stress in when we domesticate it and convert it to the cultivars or different cultivated forms through the different selection methods. Hybridization we can also carry out, mutation breeding. So these some recent, you can say some past traditional breeding methods which we are following. These method mainly focuses on development or improvement of agronomic trait. Okay, basically agronomic trait, how we are going to do the selection, by phenotype, by seeing the genetic make. We are not relying upon the genetic makeup. We are not talking about the gene level, DNA level, which we are nowadays in we have in our modern, with our modern techniques. So with this, why successful conventional breeding is, even though we know conventional breeding is the backbone, but it is a very time consuming, and sometimes most of the traits which is a quantitative one will get affected with your environment. So based on phenotype, we can't solely rely upon those traits which were highly influenced by environment during the selection. So these, in these cases, this is very sometimes it is very confusing, right? So even though many high yielding and disease resistant variety were developed, I am not telling solely now. Earlier we don't have any varieties, and when we have this boon of the modern technique, we have started releasing our variety. Earlier also we have released so many high yielding disease resistant variety which has been developed through these conventional breeding methods, which nowadays forming a base for our modern techniques, right? So now coming to this modern technique, or you can say as a this uh, non-conventional method, breeding methods, which includes, I told you, mutation breeding, tissue culture, molecular breeding, genome editing approaches. So these approaches are quite faster and more precise. Whatever is your targeted gene, targeted trait, that is only going to get affected. But in hybridization programs, sometimes we will be facing the linkage drag. Some trait is going to get linked with the undesirable trait. Okay. So tools like marker assisted selection, genomic selection, speed breeding, in which just 2 year we can cover up to F4, F6 generation, F5 generation, epigenetics and AI. So these enables breeders to handle the very complex trait, whatever, if it is tightly linked, if it is linked with some non-functional trait or non-desirable trait, that all can be get edited, or can be transformed or translated to our new variety. So together these techniques have revolutionized our crop improvement by reducing the breeding cycle. That is the first and foremost thing for release of any variety, and increasing the selection accuracy. Whatever is our desirable trait, that is going to get only chosen during with the help of these modern techniques. So I have already listed few of the techniques, so we'll be going to discuss one by one. So now coming to this QTL, this has also enabled plant breeders to create and explore different functions or functionality of gene. So during gene mapping, we have also established different backcross populations, then filial generation, double haploids, nils, rils, by using different genetic markers. And we have also different it will also provide us opportunity to target the different mixed pools or different bulk segregating analysis. And GWAS, just genome wide association study, is also one of the crucial method for discovering gene that is linked with the agronomic trait to improve the crop yield efficiently. Ultimately, whatever the objective we are confining up to, but yield we never compromise. And even though GWAS has been extensively utilized to increase yield, as well as wherever we have to confine to the single trait analysis, we can switch to this GWAS. And in pan genome can also show diversity of species, and dig out the genes which is related to important. Whenever we want to you might have been heard about the single variety that should be incorporated with different traits, means multiple traits should be incorporated in a single species. So in that case, if we want the diversification of a speciation, we should follow this pangenome. This is nothing but a sum total of all the genes in the species, which can be divided into core gene sequence, means whatever is existed in the whole species, that whole population. And another one is dispensable genome, which exists either in the single material or multiple material, which after transformation you wants to develop the new one. So with this, now coming to this, with this, I want to say that these tools, QTL mapping, GAUSS, and pangenome, this also provides a breeder's opportunity to exploit the hidden genetic variation as well as to improve the yield and stress tolerance effectively. So now coming to this genetic engineering part, genetic engineering allow the direct introduction or modification of genes which control your desirable trait. Whatever is the trait of interest, wherever you want to incorporate it, you can go with this genetic engineering process. So here it includes the different technologies like CRISPR-Cas technique, precise genome editing without introduction of any foreign DNA. And this approach has helped many breeders to develop a crop resistant towards the different biotic, abiotic stress and environmental stress, and which significantly contributing towards this food security. So now coming to the marker assisted selection. So MAS uses DNA marker, which is linked to the target trait for the early and accurate selection. You can just link the this DNA marker is linked with your targeted trait, and which will help you to go with the selection which is as early as possible, and that should be a very precise one, accurate one. It will also initiate or accelerate your breeding process, and it will also reduce your population size. But in case if you go with the conventional method, so handling the F2, F3, F4 population is very, very precise. But if you target the trait and you can screen out with the help of marker, you will reduce your population also. So that has been also reduced the bulkiness of your less material, precise selection. This is the slogan for every breeder, right? So now with this, this will also accelerate your breeding process, reduces cost, and minimizes the environmental influence. So the process includes, there are different steps. Like first, we need to identify the QTL or identify the marker. Then second step is the development of population, choice of your population, then genotyping. Then you have to go the foreground selection, what are the trait where it is present, how you are going to identify your parental lines, and what is your existing line, what is the drawback, and in which background you want to transfer that particular gene. Then you have to go for the next for the development of breeding population. Then you have to go for the pyramiding of gene. Ultimately, whatever may be the steps, like firstly you have to identify the marker and QTL. You have to validate the marker trait association in different genetic backgrounds, okay, before utilizing it. Then you have a choice of parent. You just design the crosses. You can just choose either it based on the yield or based on some different traits. Either it may be the biotic resistance, abiotic resistance, or towards some good agronomic traits. Then you can develop a breeding population, make the crosses, segregating generation. Ultimate thing, whatever is the trait is transformed. That is with phenotyping, through conventional method, it is very difficult. But when you run the marker and you can check your trait is specific trait that will give the precise, accurate, and efficient selection, which which can be only done with the help of this marker assisted selection. This can also reduce, you can say, linkage drag property. So ultimately, whatever, if the traits has been if the trait has been transformed to the from the desirable parent to the recipient parent, then before, prior to the variety release, the different field testing should be done, right? So now coming to the genomic selection. So genomic selection also predict the breeding value using genome-wide marker data without extensive phenotyping. And it also gives you opportunity for early selection at the seedling stage, which significantly reduces the breeding time. Means if the seedling stage, if you will be able to identify the trait of your interest, this will reduce your breeding time. And whatever may be the complex trait, like different multiple gene associated trait or different quantitative trait, qualitative oligogene is okay for even though you, when you want to transfer any trait or in that case. But for multiple gene or polygene trait, it's very difficult. So in that case also, genomic selection is very, very, very helpful. So when we want to go for the advanced breeding type, breeding approaches, high throughput phenotyping, like when you want to, we are not going in the field, going through some of the morphological traits, and we are just choosing the material. Here we have the here in this phenotyping process, we are utilizing different type of sensor, different types of imaging and robotics to just measure different plant traits, which in this case, what your plant is going to just move on through the conveyor belt. They can scan every, whatever may be the trait, either it may be the root parameters, shoot parameters, that can be you can see the scan and image has been provided you, the whatever may be the specific data you wants to collect related to your um, shoot data, root data, or whatever may be the plant height, whatever it may be. So this is a non-destructive method, and it also bridges the gap between your genotyping and phenotyping, which enables to precise the trait analysis for large scale, whatever may be your data. Suppose in field, if we go, what we do, we just tag the plant on 5 plant, random, not biased. We are doing random selection, we are making 5 plants, and through that we are taking the data, and we can doing the average, plant 1, plant 2, plant 3, plant 4, plant 5, of each replication, and then we can taking the average of it, right? So this conversion data collection part can be reduced with the help of this high throughput phenotyping. So now coming to this, you can say genome editing part. I have told you ki, this CRISPR-Cas method, or genome editing tools like JFNS, Talents, or CRISPR-Cas9 allow targeted gene modification. So CRISPR is the most widely used due to the simplicity and efficiency. And this also plays wherever we want to develop, or we have opportunity of developing a climate resilient crop capable of withstanding with drought, heat, flooding. In that case, this provides a very, very good opportunity, or you can say a platform. And this contributes towards ultimately getting the sustainable agriculture, fulfilling the sustainable agriculture goal and food security.
हेलो मैडम।
Hello.
हाँ मैडम।
Yeah. Sir, I am audible.
हाँ, यू
Uh,
आर ऑडिबल।
So now coming to the next slide. So here we can see this provides an outline, like how we during this CRISPR-Cas process, how we are doing the identification of putative candidate gene, which applying for genome wide association study approach. So here you can see the whatever may be the selection part, we need to go for the diverse panel or breeding population. Diversity should be there, and that not should that is not again done in a very small size of population. That should be a very big base population. So then we just go to the collection of the genotypic data and phenotypic data. And here you can see we are just using this techniques and different to identification of this putative candidate gene. We will be including the genomic data, then proteomics, metabolomics, transcriptomics. So whatever maybe means, earlier phenotypically we are just identifying the uh, you can say during the selection we are identifying the lines, but here at the gene level, even at the DNA level, RNA level, protein level, we have the opportunity to identify the each and every sequence level alleles that can be utilized here in terms of the putative candidate gene. And we can here identify the variants among that, and with that we can validate our different functional genomic sequences. Okay, so here this is also one of the example in brassica, like how what are the different steps, like how this CRISPR-Cas mediated genomic engineering. First one is the genome target. So we have target the genome sequence, and then with that we have using the small promoter RNA, we have this sequence that should which should express, and then the guide sequence will should match the target. This is the SG RNA designing, the second step. Then we have the assembly of this Cas9 construct, and then we want to transform in the Brassica napus here. So we have the three types of methods, either with the floral dip method, protoplast transfection, or first one is the agrobacterium transformation in seedling. So with this, then we, if transformation occur, then we will do it the regeneration and screening of this CRISPR-Cas9 target. So with that, we require the restriction enzyme, that assay, and then the selection of this genetically engineered specific modification line is ultimately this edited by this CRISPR-Cas. And another one thing ki this whatever the gene editing or genome editing we are this CRISPR-Cas or Cas9 facilitate, that never comes under this transgenic. Even though if you have any abiotic biotic resistance, if you want to develop any variety, biotic abiotic, just first you need to identify your candidate gene. Then only with that, with the help of that candidate gene, then only you will be able to target your, that is your targeted trait, and that is need to be get then edited with the help of this CRISPR-Cas. So mainly this genome editing part is very much reliable and helpful whenever we want to develop any, especially the traits related to abiotic stress. That is the targeted one, because these kind of disease, pest, biotic, abiotic stress is not a oligogenic. Mostly they are polygenic. So in this case, the transformation of trait without linkage drag, it's very difficult. So for that, the precise accurate part is only when we go with the genetic engineering, either with the marker assisted selection or with through this gene editing through CRISPR-Cas. So now epigenetic modification, this will also regulate the different gene expression without changing the DNA sequence. Here in epigenetic modification, only the expression of genes get altered. You are not going to change any DNA sequence. So this study has been first carried out in Arabidopsis, that has shows the genetic regulation clock gene which contribute toward the heterosis. This has been confirmed from there. Then transcriptomic changes in hybrid often enhances this stress tolerance and growth, explaining the hybrid vigor. So this has been first identified and studied in Arabidopsis. That related, this has been studies how the gene and pathway and expressions changes without any change in the DNA sequence, and how it will affect ultimately the heterosis. So now coming to the mutation breeding part, as I told you, even though we have also different, so many varieties that has been uh, we have also recovered through the natural mutation breeding. But another one thing is like in mutation breeding, this will create genetic variation, but that has been depends upon the rate of frequency. Like we have micro mutation as well as the macro mutation. So if the in case of mutation the changes is very slight, sometimes it is confusing, might be it is due to your environment. Okay, so till now 1700 mutant variety has been released worldwide, and here either the physical mutagen or chemical mutagen, especially EMS mutagenesis, have been successfully improved the yield and tolerance towards stress and quality improvement in different crops like rice, wheat, and sugarcane. So, apart from this physical mutagenesis, major and more accurate, precise variations due to mutation is visualized only in case of chemical mutagenesis, and especially this ethyl methyl sulfonate. This how this shows the substitution, means changes in your base pair. This shows a conversion. This mainly convert your purines to the pyrimidines, and this has more this mutagen EMS has more tendency, more frequency to create more number of mutants, and that too dominant mutants. What we say, the mutation mainly is a recessive one, but through this we can have the dominant mutant, so can easily identify phenotypically and expresses, right? So this EMS is most widely used mutagen in crop mutation breeding. So here we have also some of the examples in different crops, like we have developed through mutation recent new salted tolerant wheat varieties. And then we have also in bitter melons, we have also some mutant collections we have done. In sugarcane, especially vegetatively propagated plant, mutation breeding is also a kind of boon. And we have also improved in sugarcane mutants traits like resistance to black spike disease. Early maturity has been gained along with the high yield. So now coming to tDNA insertion and transposon. So tDNA is generally through agrobacterium that leads to the gene disruption and mutation, right? And transposon is, you can say, this is the first discovered in maize, and this can create a genetic variation which show the transmission or movement within the genome sequence, that within the genome. Both the tools, this tDNA insertion and transposon, these are valuable functional genomics and is very much very improved technique when it is co-jointly utilized with the uh, traditional breeding method. So here you can see there different phases has been shown, like how earlier there is a. So here you can see there is how the different stages. Here we can see a natural mutation, then that is spontaneous. Then here we comes to the physical and chemical mutagens, and then the next phase it came here, tDNA insertion and transposon method. So we have the different generation method, population, and with the help of different morphological cytogenetical markers, we are just identifying and utilizing the things and the traits for the um, to the point traits, or you can say a confined or in a precise or efficient way. So in transposome, there is one example I have collected, like there is TOS 17 mutants that is recent identified in rice variety, and through effective tissue culture method, they have transformed this mutant, and then they have regenerated it with the help of this tissue culture method. So now coming to the biparental mapping population. So whenever we want to go for the GWAS, this marker assisted selection, in that population size should very much important. So there are different biparental populations such as F2, back crosses population, double haploids, reals, nils. So this is the first and foremost crucial stage in biparental mapping approach when you go for this. So there are different steps involved in biparental mapping, such as gathering of parental genotype that should have different desirable trait. Then choosing of molecular marker with parents, like two diverse parent, diversity should be there. Then creating the mapping population. Then in the mapping population, genotyping and phenotyping should be done. Then identification of appropriate QTL. So these are the different steps. So apart from that, there is also not only biparental, multi-parental mapping is also there. So in that case, multi-parental populations like NAM, nested associated association mapping, or MAGIC, that is multi-parent advanced generation in interprocess. So especially these biparental and multi-parent mapping is very much important when you are approaching towards this modern techniques. So here we can see that there is this one experiment which was conducted by Yuval et al, which has proposed about the NAM for analyzing the genetic architecture for the maize flowering time. In that 5,000 reels is there, started from the, then from 25 families. Then it, here you can see the how the progress is going to. It's like from the 500 baselines, it is going to get reduced into the smaller size population till they have selected the reference line for this uh, screening of the trait or objective is they are confining to the flowering time, less flowering time, early maturity. So with that, there are different traits which can be mapped, like disease resistance, leaf architecture, in maize by using this NAM approach. And Arabidopsis thaliana was used to create the first magic population in crop. That's why we called it as a Drosophila of plant kingdom. Right? So now coming to the speed breeding. This is the speed breeding is nothing but a controlled environment, like how the breeder is screening their material in natural environment. Artificially, we are creating by manipulating the environment through different fluctuations of light, temperature, and at a different plant age can be harvested, and we can also obtain 3 to 6 plus generation per year instead of 1 to 2 in field. Either if we have the maize like crop, we can go for the three crop cycle per year. But for different crops, if have opportunity for summer kharif or either kharif rabi. So now in this we can integrate our marker assisted selection, genomic selection with this our speed breeding technique. So in that we can rapidly fix our desirable allele, whatever may be the crop we can approach it. Either it may be the wheat, barley, rice, the work is going on. So first and foremost thing in this most utmost thing, with the help of this speed breeding, we can rapidly develop the improved variety, and this can significantly shorten the breeding cycle. So here we have the small setup of 96 cell tray. In that we can uh, regulate the photo period or light intensity as per the short day crop or long day crop. Okay, so in the case of short day crop, we are considering here soybean, rice, and amaranthus. And temperature, it should be typically for wheat protocol, it is 22 degrees Celsius day, and in night 17 degree, and humidity it should be. So whatever is your, which this will accelerate the vegetative growth and flowering without providing any kind of stress. But in normal condition, now if you have the wheat in your field, how much chilling and fridging stress it is offering, and it is perceiving. So in this is the good way of screening the material in speed breeding. In even if in this case your trial never failed, but in the field, due to the fluctuations of the temperature, dropping of the temperature, this will affect the vegetative growth, the flowering period, or grain filling time. We can maintain whatever maybe the wherever the whenever the water is required, irrigation, proper nutrient, light, proper temperature, humidity, everything is managed during and with the proper sufficient time frame, and we can rapidify our generations. And lastly, the artificial intelligence and machine learning, this analyze large data set, like whenever ultimately whatever the selection you are doing, whatever the crossing hybridization program you are doing, ultimately you have to speak up based on your data what you have collected, and that should get analyzed. So in that case, if manually if you are taking, it is very cumbersome. But when you will include this with machine learning AI method, in that you have the lots of data. So in that case, if when you have more number of data, either that may be the targeted, that is supervised one you are knowing, and some cases your data is non-targeted. So in that case you will be finding these help from this machine learning or AI or deep AI, right? And this is a very helpful while processing of data. Here you can see how with the different approaches, like a different diversity of like how we are collecting, how we are mapping the population, and ultimately with the help of where each and every step Either you are using the GWAS or you are using the marker assisted selection, or whenever you will be having the speed breeding or phenotyping. So in that case, AI is involved or included. So modern crop improvement integrated with the conventional breeding or with advanced molecular genomic or digital technology, this will provide or enhance the efficiency, precision, and sustainability. By adopting these techniques, we can develop resilient, high-yielding crop to meet the future agriculture challenges. So, thank you.
Uh, Thank you so much, madam. And we are excited to continue our lecture series with Dr. Sweta. She is assistant professor at School of Allied Healthcare and Science, Jain deemed to be University. Dr. Sweta will be discussing the role of genetic engineering in crop improvement. So, Dr. Sweta, thank you for joining us today. Thank you.
Thank you, sir. I'll go ahead with this session. Just let me know whether my screen is visible or not. Is it visible?
Yeah, yes, madam, it is visible.